Today, more than 8 billion humans live on our planet. But if we could look back between 800,000 and 900,000 years ago, the picture would have been shockingly different. New scientific findings are shedding light on our ancient ancestors and pointing to a critical and previously unknown chapter in human evolutionary history. According to recent research, early humans, including Homo erectus, nearly went extinct less than 1 million years ago, likely due to extreme ice age conditions. Genetic evidence reveals that between 930,000 and 83,000 years ago, the ancestors of modern humans experienced a catastrophic population bottleneck. During this period, humanity lost an estimated 98.7% of its breeding population. Remarkably, our forefathers stood on the very edge of extinction. The surviving population was so small that extinction would have been almost inevitable, if not for sheer resilience, adaptation, and perhaps a measure of luck. A study titled Genomic Inference of a Severe Human Bottleneck During the Early to Middle Pleistocene Transition demonstrates just how dire this period was. The evidence shows that human population remained extremely small, yet stable, for over 100,000 years. This prolonged bottleneck may have played a key role in shaping the divergence of modern humans, Neanderthals, and the mysterious Denisovans. Understanding ancient population size is essential for reconstructing human evolution, yet the history of early human populations during the Pleistocene is notoriously difficult to unravel. To overcome this challenge, scientists developed a fast, infinitesimal time coalescent process, allowing them to calculate composite likelihoods from 3,154 modern human genomes. Using this approach, researchers reconstructed past population sizes and uncovered a dramatic decline. Their findings revealed that between 930,000 and 83,000 years ago, human ancestors experienced a severe bottleneck, with an average of only 1,280 breeding individuals worldwide. This bottleneck lasted approximately 117,000 years and nearly erased the human lineage entirely. Significantly, this period aligns with a major gap in the African and Eurasian fossil record, likely because there were simply too few humans alive to leave behind many traces. The researchers detected a population reduction from roughly 100,000 individuals to just over 1,300, coinciding with major climate shifts. These changes included global cooling, expanding glaciers, falling ocean surface temperatures, and prolonged droughts across Africa and Eurasia. Scientists are still uncertain exactly how these environmental changes affected early humans, as fossils and artifacts from this time are exceptionally rare, likely a direct consequence of the population collapse. Previous research suggests that the last common ancestor of modern humans, Neanderthals and Denisovans, lived between 765,000 and 550,000 years ago, shortly after this newly identified bottleneck. This timing raises a compelling possibility. The near-extinction event may have directly contributed to the divergence of these three human lineages. If the last common ancestor lived during or just after the bottleneck, the crisis may have split humanity into tiny, isolated groups. Over time, genetic differences between these survivors could have accumulated, eventually forming three distinct populations. Further evidence supports this idea. Earlier research suggests that two ancestral chromosomes fused between 900,000 and 740,000 years ago, forming what we now know as human chromosome 2. Because this fusion coincides with the bottleneck, scientists believe the near eradication of early humans may be linked to this major genomic transformation. Researchers reconstructed evolutionary distances among the great apes, the hominin family, using statistical outputs from their algorithm. These findings offer new insight into chromosome fusion events and provide a novel computational approach for estimating speciation timelines. The end-to-end fusion of two ancestral primate chromosomes reduced the chromosome count from 48 in great apes to 46 in modern humans. Genetic evidence for this fusion includes inverted telomeric repeats at the fusion site and degenerated satellite DNA, marking the remains of an ancestral centromere. Earlier estimates placed this fusion around 4.5 million years ago, but using an improved algorithm that detects locally overrepresented weak to strong substitutions, scientists now estimate the fusion occurred around 900,000 years ago, with a 95% confidence interval of 400,000 to 1.5 million years ago. Because Neanderthals and Denisovans also share this fused chromosome, it must have occurred before their lineages split from modern humans, around 800,000 years ago. This new analytical technique could eventually be applied to Neanderthal and Denisovan genomes, potentially revealing whether they too experienced severe bottlenecks after diverging from our lineage. In related research, another recent study shows that human skin tone has varied widely for at least the past 900,000 years. Genetic analysis of pigmentation-linked variants across diverse African populations reveals a far more complex history than previously believed. Some darker skin tones evolved relatively recently from older, paler genetic variants. For 8 key pigmentation sites, both light and dark variants existed at least 270,000 years ago, and 4 of the lighter variants date back over 900,000 years. These findings show that lighter skin variants predate modern humans and have persisted in parts of Africa ever since. Today, skin pigmentation across sub-Saharan Africa remains extraordinarily diverse. For example, the San hunter-gatherer populations of southern Africa, among the oldest branches of the Homo sapiens family tree, possess relatively light skin pigmentation. Perhaps most intriguing, some ancestral light skin alleles are shared by the San and archaic hominins such as Neanderthals and Denisovans. This suggests a shared common ancestry for these traits, predating the split of the 3 lineages around 900,000 years ago. Another groundbreaking study published in 2020 revealed that over 700,000 years ago, Neanderthals and Denisovans interbred with a previously unknown population, so-called super-archaic humans, who split from other human lineages around 2 million years ago. By analyzing how mutations are shared between modern Africans, Europeans, Neanderthals, and Denisovans, researchers identified 5 separate interbreeding events, including one that had never been detected before. This newly discovered episode involved interbreeding between Neanderthal Denisovan ancestors and a super-archaic population that had been separated from them for over 1 million years, making it the most genetically distant interbreeding event ever identified among humans. In comparison, when modern humans interbred with Neanderthals around 50,000 years ago, the 2 populations had been separated for roughly 750,000 years. These findings offer rare insight into how long reproductive isolation takes to develop in the human lineage. Using additional genomic clues, scientists estimated the timing and population size of these ancient splits. They believe super-archaic humans became a distinct species around 2 million years ago, consistent with fossil evidence from Eurasia dating to 1.85 million years ago. Researchers also proposed 3 major waves of human migration into Eurasia. The first occurred about 2 million years ago, when super-archaic humans migrated and established large populations. The second took place around 700,000 years ago, when Neanderthal Denisovan ancestors expanded into Eurasia and interbred with these earlier settlers. Finally, around 50,000 years ago, modern humans spread into Eurasia, interbreeding with Neanderthals and Denisovans. Together, these discoveries paint a dramatic picture of survival, adaptation, and shared ancestry, reminding us how close humanity once came to disappearing entirely. As we continue exploring the mysteries of our deep past, remember to embrace the uniqueness of our shared human heritage. Thank you for watching and for subscribing.